Hey everybody. So in this video, I would like to walk all of us through the process of bridge amplification that is used with some forms of Illumina technology for second generation sequencing. So if you remember in group activity 4, we left off with constructing the DNA library that is needed for the sequencing process. And if we went through that process with the NEB Next Ultra DNA library prep kit for Illumina sequencing. And you might remember that what this library prep method did was it added sequences. Now these right here, this represents our genomic DNA fragments of about 500 base pairs long. And then we came up with these sort of generic names for the sequences. So we have unique 2 up here, unique 2 complement down there. We have P7 complement here, P7 sequence here, P5 sequence here, P5 complement there. Now, I thought it might be helpful to show you what these sequences actually look like. And recall that with Illumina next-gen sequencing, we're going to have these oligonucleotides stuck to the flow cell. One is called the P5 and the other is called the P7. So I wanted to show you what the molecules might actually look like at the sequence level. So what we're looking at down here, what I just circled, is sort of the actual base pairs that you would see in the molecules in the DNA library after they have been prepared with the NEB Next Ultra NGS library prep kit. So these things right here, these ends, these are representing our DNA fragments. So one DNA fragment in our library, about 500 base pairs long. And you can match up the colors. You can see this right here is the P5 sequence. This right here is the complementary sequence to the P5 sequence, and so on and so forth. Now these T's and A's, they're going to be essentially, these represent the T's, this represents the A's, and those are found in both sort of the unique 1 and unique 2 sequences here. So, okay, so I want to walk through the bridge amplification process with this molecule down here at the DNA level, because I think it might help you see how this process works. So the first thing that's going to happen is essentially the molecules in the DNA library. Keep in mind, we have hundreds of thousands of these, maybe millions of these molecules in our DNA library, each holding a different fragment between the adapter sequences. So the molecules get denatured, and here we're just looking at one molecule. So the molecules are denatured. We have one here, and we have the other strand down here. We're looking at one molecule that's been denatured. So we have the 2 single strands. And all of the molecules, after they're denatured, they get added to the flow cell. And now they're free to anneal to the oligonucleotides on the flow cell. So in the next diagram, what I'm going to show you is we're just taking one of these, either the top one or the bottom one. And we're just looking at what one of these strands of DNA is doing as it's flowing over the flow cell. So we're going with this strand right here. And so that other strand, what happened to that? Well, that's hovering around a different part of the flow cell. And it's going to do the same thing, but in this part of the flow cell, well, this strand is going to do something interesting. So we can look at the DNA sequences here and keep in mind that this is much longer, right, that I'm actually showing here. These ends represent 500 nucleotides of the DNA we want to sequence. But look at the DNA molecules, look at the sequence of all these other sections of this DNA. And then compare these to the oligonucleotides that are attached to the flow cell down here. So we have two types. So this is the P5 oligonucleotide. This is the P7, and so on and so forth. P5, P7, P7, P5, and we're going to have thousands of these oligonucleotides in this vicinity of the flow cell. And they're all attached to the flow cell at their 5' ends, so they're stuck to the flow cell. Now, if you look at the sequences of the oligonucleotides and the sequence of this single-stranded DNA molecule from the library that's hovering above them, you might be able to see that only some of the sequences are complementary. So long story short, it's going to be, let's see, this one right here is going to be able to stick to the P5 oligonucleotide. So this one right here, this cannot stick to the P7 oligonucleotide because it's the same exact sequence. It's the same sequence. So they're not complementary, they're identical. So this part of this DNA strand is going to stick to one of the P5 oligonucleotides in the vicinity of this flow cell. I have that diagrammed like that. Okay, so this is that DNA molecule that I showed you in the previous slide, and now it's annealing to one of the P5 oligonucleotides. It could have annealed here or here, but random chance, it annealed there. So, and this is happening all over the flow cell, right? These single-stranded DNA molecules from our library are annealing either to the P7 oligonucleotide or the P5 oligonucleotide, depending on what strand of the molecule from the DNA library was found near the oligonucleotides. Okay, so now what happens is the polymerase is added, deoxyribonucleic acids are added, and we have synthesis from the 3' end. So this strand is going to be copied by DNA polymerase. And we're going to end up with a double-stranded molecule at that part of the slide. But note this 3' end down here, that 3' end, it's not covalently linked. It's not covalently linked to the flow cell. So after it's copied, as I'm showing here, all the DNA molecules are denatured again, and the only things that remain stuck to the flow cell are those that are covalently linked as a 5' end. So this whole molecule now, this strand will stick, but after we break those hydrogen bonds by denaturation up here, this one is going to float away and be washed off the flow cell. So here I have it floating away because the DNA was denatured. But this one right here, this sticks to that oligonucleotide. This sticks to the flow cell because it's stuck to the flow cell at its 5' end. Now, okay, what happens next? So essentially, the conditions of the flow cell are changed or altered in such a way to allow annealing to occur between DNA molecules where they're complementary. And so if we look at this sequence right here, we will now see that this sequence is complementary to the P7 oligonucleotides. So it can anneal to here, to here, or to here. So it's sort of random chance which one it anneals to. So what we have, it's annealing over here. Okay, great. So what happens next? Okay, DNA polymerase is added to the environment of the flow cell, deoxyribonucleic acids, and this is the 3' end. So synthesis occurs in this direction. And that DNA molecule is copied. So, and that's sort of the bridge. That's how we get this bridge amplification. You can see this bridge forming, right? So that's pretty neat. Now, okay, again, we have another after the replication has finished, the parameters of the replication are such that the polymerase has time to finish, but then the DNA is denatured again. So the conditions are changed in the environment of the flow cell to break those hydrogen bonds between the DNA molecules. And now what we have, but neither of these is going to float away now, right? Even when the hydrogen bonds are broken between these two molecules, neither is going to float away because they're both attached at their 5' ends to the flow cell. So there I have both strands copied. And now after denaturation, okay, well, the strands are sort of, free flowing in the flow cell. Now, again, the conditions of the environment of the flow cell are changed to allow complementary base pairing. And what can happen is see how this sequence right here is now complementary to the P5 oligonucleotides. And this sequence right here is complementary to the P7 oligonucleotides. So the process can happen again. And it's getting really complicated for me to diagram multiple cycles of this. So what I'm going to do is I'm going to change all the red with the letters and the green with the letters and the yellow with the letters to just a red and green and yellow sort of lines. So you can see that here. So again, conditions change. So these are going to be able to anneal. These ends are going to be able to anneal to the P5 and P7 oligonucleotides. And here we have them annealing. And so we can get extension this way from the 3' end, extension this way. Okay, now keep in mind, these 3' ends are not attached. So when we denature, they're going to be free flowing in the flow cell, and the molecules will be stuck at their 5' end. So I'm showing the DNA being copied there. And okay, denaturation. And now we have all these strands just sort of free flowing, but they're still stuck at their 5' ends. Okay, so we do many, many, many cycles of that. And so we end up with something like this. Of course, there are hundreds more. I didn't have time to diagram all of them, right? But there are hundreds more of those there. And I have these out of order. So we end up with something like this. So imagine the last denaturation step. Okay, so all of the 3' ends are free. All of the 5' ends are stuck. This is one cluster on the flow cell. We can have millions of these clusters. This is another one over here. Each cluster has its own genomic DNA fragment. The genomic DNA fragments are identical for all the molecules in the cluster. And so what happens next is the 3' ends are blocked. Let's see, how did I do this? Two things happen. One, the molecules that are stuck to the flow cell at the P7 5' end right here, these are released. These are released, and I'm not really sure how that is done. I don't know if it is proprietary, the particular process used for doing that, but the sequences are cleaved and they are washed off. So we're stuck with these P7 oligonucleotides here. And the 3' ends, after that, any free 3' end is blocked. Any free. So that's what these little black things are. They're blocking them, so they can't serve as, say, primers during the sequencing step. So they can't be serve as a primer that the polymerase can add a nucleotide to. So I think that is it. Yes. Okay. So now we're ready for the sequencing. And essentially, the sequencing primer is going to bind right here on each of these molecules. And the arrow is where the 3' end is. And this gray sequence right here is this gray one right here. I have 3 different gray segments in each DNA molecule, but the middle one is the genomic, represents the genomic DNA fragment if we're doing genomic DNA sequencing. And they're all identical. They're all identical. And so now we're ready for the sequencing reaction, the sequencing by synthesis process. Okay, so I hope that helps you understand how the bridge amplification process works. If not, please send me an email or come to office hours and I can go over it in a different way for you. Okay, see you in class on Wednesday. As soon as I figure out how to stop this recording.